Hi all, this is Dr. Fry. I'm here in my kitchen with some bean seeds. These are ones that I have soaked for a few days in water just to soften the seed coat, which is on the outside here. You can see it's a little bit shriveled. I just took it out of the water. This one is definitely softened. So this is the seed coat. And if you recall from our notes, the seed coat developed from layers of cells called the integuments. Okay, so that developed from that. There's a little bit of additional external anatomy we ought to be familiar with here. So I'm going to point out this is the oval hilum, this pale oval here. This is where the ovule had attached to the carpel, which is part of our ovary. Okay, so this is where the ovule, which led to the seed, developed into the seed, was originally attached to the ovary, this point here at the oval hilum. There's another point here I want to point out, and that is the micropyle scar. The micropyle scar is the point that allowed sperm to enter and fertilize the egg. So this is where, if you remember your tube cell, it produced a pollen tube which grew down the stigma style and into the ovary and allowed the sperm to enter and meet with the egg and the two polar nuclei. Okay, so at that point there, micropyle scar. Alright, so that's about all the external anatomy you need to be familiar with. We're going to take a seed here and just remove the seed coat. And like I said, these have been softened for a couple days in water, so that seed coat will come off pretty readily here. Just if you dig your fingernail in, you can easily peel that back. You can try this at home. But this will expose our cotyledons. So this is a dicot, and so it will have two. And as you can see here, there are two. They separated pretty easily. We can just open that up entirely and show you the inside. We've got two cotyledons. They are now taking on a storage role for this seed. It's got starchy substances here which will feed the plant. And for dicot that has epigeal germination, these will also grow out from the ground and for a short period of time undergo photosynthesis to allow the plant to get started producing its own sugars. Alright, so I'll just point out now, you can see that there's a couple little structures here. This is the embryonic axis of the plant. You can see there's a tip here. That is the radicle. The radicle will become the root of the plant. It extends upward to the hypocotyl. That will become the shoot of the plant that is below the attachment point of the cotyledons. Okay? And then just above that is the epicotyl, which is above the attachment point of the cotyledons. And that leads to some very fine little leaves. I'm going to try to show you those here. I'm going to stick my toothpick underneath. You can see there are some little leaves here which are called plumular or foliage leaves. Those will be then the first recognizable set of leaves on the seedling as it grows up. So that's about all the anatomy you need to know for this seed. We've got our plumular and foliage leaves, an epicotyl, hypocotyl, radicle, one cotyledon, and the other cotyledon, the seed coat, and then the structures on the outside of the seed, the oval hilum and the micropyle scar. All right, thanks for listening.